here I actually, the people who did the work, I will refer quite a bit to experimental work as well. That was done, as I said, in the levels 1 with Dogic by a current postdoc, Itamar Colvin, student, Ray Atkins, and the undergraduate, Sven Witthaus, that just actually left for graduate school. And the theory work that I will describe was done by two postdocs in my group, Jihong Yu and Fernando Caballero, and the student Parth Gulati. And so what I will do to start with is sort of introduce both the topics of my talk, tell you a little bit about active matter and about interfaces, and then put them together, and we'll see what activity does to interfaces. But before I start, I actually want to thank not just IIT Chennai, IIT Madras, but actually the entire local community for sending me some truly exceptional students over the years, and in particular that actually have contributed not directly to the work I will talk about today, but have contributed to my work in active matter. And they are Suraj Shankar, who actually graduated from IIT Madras in 2014, and is now a junior fellow at Harvard. And Shiladitya Banerjee, who actually graduated from Chennai Mathematical Institute in 2008, and now is an assistant professor at Carnegie Mellon University in the US. So I'll be very happy to get any more students from either institution. They were both really wonderful, great scientists and great people. Okay, but let's start with interfaces. As you know, immiscible fluids, such as oil and water, phase separate, creating soft fluid-fluid interfaces. And the physics of liquid-liquid phase separation has applications to many fields, from cooking, making salad dressing, to materials design, the creation of, for instance, these block copolymer structures shown here, and also in biology, where it drives the condensation of proteins into membrane-free organelles, as shown in this video by the lab of Brian Nguyen at Princeton, where what you see is something called granules, these sort of yellow-green spots in C. elegans that are membrane-free structures. So they are liquid drops, essentially, of protein implicated in controlling cell fate. And so this has motivated us to ask the question of how active processes affect interfaces and liquid-liquid phase separation. But before I tell you about that, let me spend a few minutes telling you what active systems actually are. So sort of a 2 minutes introduction to active matter. Active particles, you can think of them as engines that actually consume energy to generate their own motion and forces. An example is a bacterium that uses food to swim or to crawl on a substrate. And there are, of course, also synthetic microswimmers that use a variety of phoretic effects to become self-propelled particles. Active matter is a collection of such particles or engines interacting with each other, and collectively they organize in a variety of structures on many scales. And there are examples from both the living and non-living world, from structures composed of a variety of protein inside the cell that, for instance, drive the separation of genetic material, to, as I said, engineered microswimmers that we call active colloids, to bacteria, eukaryotic cells, miniature robots, and even at our scale, a collection of fish, birds, or even human crowds. So what all these systems have in common is that the individual, there is an energy input at the scale of the individual, so sort of the micro scale, and this energy cascades to drive dynamics and organization at large scales. For instance, protein like myosins inside cells transform chemical energy into forces to control how we actually move and function. Now, let me give you a more specific example from biology. What you see in this image is a structure that you find inside the cell, composed of long biofilaments, long polymers called the microtubules. And it's a sort of a network where these polymers are cross-linked by motor protein, particular a motor protein called kinesin. This is actually a kinesin cluster. It's supposed to be a cartoon of a kinesin cluster. And these protein essentially are able to convert the chemical energy into mechanical forces. And by walking literally on these filaments, they displacement relative to each other, as indicated by these arrows, driving motions inside, for instance, in this case, inside the cell. And this is actually a video that shows how the structure called actually the mitotic spindle organizes and segregates actually genetic material upon cell division. This is a video from the lab of Wadsworth Lab at UMass Amherst. And so the idea is that, again, you have these sort of microscale machines that organize the spindle, the structure of microtubules, and ultimately rearrange the skeleton of the whole cell and drive cell division in this case. The reason I specifically mentioned this example, a collection of microtubules and kinesin inside living cells, is the following. And by the way, I should have said at the beginning that please do feel free to interrupt and ask me questions at any time. Actually, that's very helpful, so I don't feel I'm speaking just to the vacuum. So just pick up if you have a question and interrupt me at any time. So these are precisely the ingredients that, starting back now 10 years ago, the lab of Zvonimir Dodig used to assemble an in vitro active fluid, or actually active liquid crystal, which is essentially a mixture of this microtubule and kinesin powered by ATP, the fuel that the microtubule use to undergo to generate the chemical energy that they transform in mechanical forces. And these are always two-dimensional examples, meaning this suspension of microtubules and kinesin is actually sitting at an oil-water interface. At the level of when the filament density is low, as you see in this video here, what you see indeed is that you have the strict bundles actually of microtubules cross-linked by kinesin, and the kinesin drives the microtubule to move relative to each other, and so the bundles split apart, slide, and the system is continuously reconfigured. At higher microtubule density, essentially you get an active gel, where the energy generated by the kinesin motor drives both local alignment of the microtubule and this sort of self-sustained turbulent dynamics powered by the motors. And this dynamic survives for as long as this ATP is present in the system. Now, to just understand a little better what drives these dynamics is indeed driven by these forced dipoles that are generated on by microtubule bundles through this kinesin. And these forced dipoles push outside on the surrounding fluid and drive the motion. So they generate what we call active stresses, which of course are controlled by, which direction is controlled by the orientation of this filament. And here is the expression for the active stresses generated by these bundles. They're actually proportional to a quantity called Q, which is the order parameter for the alignment of or nematic order of these filaments. This quantity S is the strength of alignment, how alignment the filament are, and this unit vector N, called the director, is the direction of alignment. The coefficient in front, alpha, we call it activity, and depends on the specific biomolecular processes that drive this motion. So in this case, it will depend on the concentration of kinesin and on the concentration of KITP. And by the way, this formula for this active stress was first proposed and its consequences worked out on driving this motion in a paper about 20 years ago by Aditi Sima and Sriram Ramaswamy. This coefficient alpha can be both positive or negative depending on whether these forced dipoles point out or push out or pull in. The systems I'm interested in, these active gels or active nematic, as we call them, have a negative alpha, which corresponds to extensile forces or forced dipoles that sort of push outwards. So that's my brief introduction to active matter. This is the kind of active system we will use. And what we really want to do is sort of put it in contact with a passive fluid and study the interface between the two. And again, the motivation is that we know that in passive fluid, like again here, oil and water, we can actually, they would phase separate. Here, water is at the bottom, oil is on top. But if we shake the container, that is put energy in, inject energy at the macroscopic scale, we kind of restart the phase separation. And therefore, the energy we inject at large scale cascades to small scales. Now, what we like to ask is how instead energy injected at the small scale to the kind of active processes I showed you affects interface, this kind of interfaces and liquid-liquid phase separation. Are there any questions so far?
So
okay.
The previous example,
yeah, go ahead.
Yeah, in the previous example, it is a 2D or a 3D system?
That was 2D.
That is also 2D.
That was 2D, that example. And I will tell you the geometry of the examples. They're going to be essentially always thin films, the examples I'm going to show, although they will have a finite thickness of about 60 micron, actually, 60 to 80 micron. Okay. Anything else? Okay, so after that introduction, what I want to do then with the rest of the talk is tell you, first of all, about how the Dogic lab was able to design essentially active-passive fluid mixtures that allow you to investigate the role of activity on interfacial phenomena. I will then tell you how we construct a continuum model to try to describe these interfacial phenomena, in particular interfacial fluctuations, surface waves, and active phenomena. We are also studying the coarsening in active phase separation, but I probably will not have time to tell you about that. So let me start by showing you, first of all, the experimental system that they were able to actually construct. So what I did is to take a passive phase separating polymer mixture, specifically a mixture of PEG, polyethylene glycol, and dextran. These are two polymers that phase separate in equilibrium. And so if you start in this region of the phase diagram, they will spontaneously separate into a PEG-rich phase and the dextran-rich phase. And the coarsening is driven by interfacial curvature of the various drops and coalescence. And this is just equilibrium coarsening of this system. Then what they did is that they mixed up with this polymer mixture their microtubule and kinesin. The microtubule are fluorescently labeled in red, and they also labeled in blue the dextran. And they were able to then show that when you put in microtubules and kinesin, the microtubule completely segregate, I should say, to the dextran phase. So now you still have this mixture of dextran and PEG. Here the dextran is blue, and the microtubules are in the dextran. So this microtubule dextran system becomes an active fluid in the presence of ATP. And the PEG is the passive fluid. And the key property is that this system, this mixture, has a very low surface tension, very low interfacial tension that matches the magnitude of the active stresses created by this kinesin motor in the active fluid. It's a very small surface tension. It's 4 orders of magnitude smaller than oil and water. And then, so now if you put this mixture into a vertical cell, so this is a thin cell about 60 micron thick, what happens is that the active fluid is actually heavier, so it goes to the bottom in the presence of gravity, and they actually undergo centrifugation to have the active fluid sediment. And then you can watch the interfacial fluctuations. And they're actually visible by eye. The scales you see here is millimeters, so this is a really big system. And these are very large fluctuations of the interface. So this is the active fluid at the bottom and passive fluid at the top, and those are the fluctuations of the interface. And temperature doesn't play any role here. Well, I'll show you that in a minute. So the size of this fluctuation is surprising for the following reason. Let's think a minute, remind ourselves how temperature drives fluctuations of liquid surfaces. So say you have the surface of a liquid, you can image it by a scattering experiment and look at the interfacial fluctuations. You can actually calculate the size, the spectrum of interfacial fluctuations very simply in the following way. So you have a liquid of density rho, say there is just vacuum above, and it has a surface tension sigma. You start a flat interface, then a deformation can be described by a height h, how high the interface is above the flat reference state. And the energy cost for such fluctuations will be proportional to the surface tension and gradients of h squared. The dynamics of fluctuation can be written as controlled by sort of a Langevin equation. I've also added gravity here, and this is just thermal noise. And there is a characteristic line scale in the problem that comes from the interplay of tension and gravitational forces, known as the capillary length. So if you calculate the spectrum, this is in Fourier space, a Fourier transform with respect to x, the position along the interface. Calculate the spectrum of the interface. These are driven by temperature, so their strength is kT. And a very small wave numbers, they're flat because gravity dominates, and there's large wave number, they decay as 1 over k squared. This is a typical spectrum of fluctuations. If you use this to estimate how much temperature will roughen the interface of my active passive fluid, so for this peg-dextran interface, actually in the absence of activity, I should say, you will find that the variance of the fluctuations of the interface, you can put in number and estimate it, is of order of 100 nanometers. This is about the size of dextran molecules, certainly not something visible by eye. So these large fluctuations that you see here, now here this video shows 3 videos actually for increasing concentration of kinesin, KSA stands for kinesin protein that drive activity. These very large fluctuations are really driven by activity. And we like to understand how activity can drive these large fluctuations and try to quantify this observation in a quantitative way to describe this observation. Any questions about the experiments?
Hi. So can you hear me?
Yes.
So I have a question, like when we are increasing this kinases, so we see like there is an increase in the activity. So what are we
you're very choppy. Say it again.
Yeah, I'm telling like when we increase this kinases, we see like there is an increase in the activity.
Yes.
So like what about the time it takes to phase separate? Like
honestly, I'm not sure. This is all after the system. So they centrifugate the system, actually. See, the cell is vertical, and they centrifugate to get the active fluid to go to the bottom because it's heavier. Mm-hmm. And then once you have, and that's how you get it to actually phase separate this way. And and then they measure this interface. There are some time stamps here. These are minutes and seconds, but these are the time for the fluctuations, not for the phase separations. I think for actually, they also do experiments that I will not get to in an horizontal geometry where the system coarsens on its own, and then it's a matter of hours.
Okay, thank you.
Okay, but yes, absolutely. So here, increasing kinesin concentration increases, from my point of view as a theorist, this, let's see if I can find it again, increases this parameter I called alpha, which I will call activity, and drives these fluctuations. So it's not temperature, but are these active stresses that drive the fluctuations. And now what I would like to do is try to show you how. Okay? Okay, so let's try to develop a continuum theory of these active interfacial fluctuations. Okay, now, first of all, I want to spend a minute reminding you how we actually, over the years, have constructed a hydrodynamic theory of these kind of active liquid crystals. So I said that These are suspensions of elongated things, microtubules. There is a lot of local orientational order. So to describe the dynamics of this system at the continuum levels, we are going to use two continuum fields. One is the flow velocity. We'll have a sort of a Navier-Stokes type equation for the flow velocity. And the other is this orientational order parameter, sometimes called the Q tensor, which is a measure of local alignment of the microtubules, okay? And it's a tensor because the order here is not like in a ferromagnet where it has a direction, it only has an orientation. So, you know, up and down is the same in two dimensions as far as this order goes. So we construct equation for these two fields. Because these systems really are on a substrate, so there is friction and they are generally at very low Reynolds number. Instead of a Navier-Stokes equation, we really write a Stokes equation, neglect inertia, where simply balances all forces on a fluid element. And so this is the balance of friction with the substrate, or with the walls in this case, because it's a vertical cell, so we should think about the walls. There's viscous stresses, pressure gradient, and then we have the gradient of two stresses here. These active stresses I introduced earlier, which are proportional to activity and to the nematic tensor, and elastic stresses that arise from these also nematic degrees of freedom, which describe the energy cost associated with deforming the local orientation. Flows deform the orientation, and there is an energy cost to that, or if you like, the formation of the orientation generate flow. There is an interplay between these two. So that's the equation for the flow velocity. Then we have an equation for the dynamics of the order parameter Q that essentially says that the rate of change of Q is determined by relaxation. Q relaxes with some time scale, and this psi square is proportional to the stiffness K, is the length scale again over which the order parameter is deformed spatially. And then there are terms that couple the order parameter to flow. There are terms that say, so this is the strain rate, and they say that shear flows tend to locally align the filament, and so shear flows generate local active order, local nematic order. And vorticity, omega is the curl of V, is the vorticity of the flow. Vorticity can change the direction of the order, and so there is this kind of coupling to vorticity. So this is hydrodynamics of any active liquid crystal. There is no mixture here. There is only one fluid for now. And it says the dynamics of the system is controlled by the interplay of flow and alignment, with shear flows generating alignment and active stresses proportional to alignment generating flows. And this has been studied a lot, but what we want to do now is to sort of generalize this to describe an active-passive mixture. So to do that, and this is also a fairly well-established model, I should say, to do that, we introduce a phase field, which is simply advected by flow. Actually, this is not supposed to be a time, there's no time derivative here, just a bad cut and paste job. This is just gradient of phi. So we introduce a phase field phi that is 0 in the Newtonian fluid and 1 in the active fluid, and this is advected by a flow. So this phase field is really just a way of keeping track of our two fluids, if you like. And then we write again our hydrodynamic equations, but now notice that, first of all, these active and elastic stresses that couple to the liquid crystalline degrees of freedom are only present in the active fluid where phi is 1. And in addition, we add to the flow equation capillary forces that arise from deformation of the interface, and therefore are proportional to the interface curvature, which depends on gradient of phi, and to gradients of phi itself, as well as to the surface tension. So these are the passive restoring forces that, upon fluctuation of the interface, tend to restore a flat interface. So this is sort of a closely related to what people call multi-phase field model. It's a bit different because we don't include, we want a very thin interface, so we don't allow for diffusion in the interfacial region. That's why there's a 0 on this side of this equation. And we solve this numerically using something called open form. And when we do this, if we compare, so here are our simulations and here are the experiments, we see a very similar dynamic. So here's the active fluid, above is the passive fluid, which, and here is the interface. And what you see is a lot of local alignment of the filaments, and essentially these active stresses that generate these turbulent-like flows in the bulk push up the interface and generate these strong fluctuations. We can control the spectrum, we can calculate the spectrum of the interfacial fluctuations, and they are qualitatively at least similar in simulations and experiments, so it's hard to make a quantitative comparison. But what is certainly clear is that as we increase activity, these curves are increasing that parameter alpha in the simulation and increasing the concentration of kinesins in the experiments. As we increase activity, the strength of the fluctuations becomes larger and larger. Okay, let's try to understand that a little better. So, we simulated the equation numerically, but we can actually do something more. We can actually derive, at least for the case where for small fluctuations to linear order, we can derive an equation for the high fluctuations. The rate of change of the height is simply the velocity, the vertical component of the velocity at the interface. So that's what pushes up the interface is the flow, and tension then tends to restore the flat interface. So to calculate this velocity, we solve the coupled equations for the coupled Stokes equation for the Newtonian fluid at the top and the active fluid at the bottom. We actually neglect the elastic stresses, so we only have active stresses here. These are incompressible fluids. We solve these coupled equations with boundary conditions that XY stresses are continuous, but there is a jump in normal stresses determined by gravity and by surface tension. When we do this, we find that the expressions are fairly complicated, as you will see, but essentially we can split this velocity in two parts. What I call a passive part, which really comes from, if we neglect viscous stresses, only incorporate friction for simplicity. We have calculated the full expression, but I didn't want to write them here. This relaxation is just like in equilibrium, in passive system, controlled by surface tension and by gravity, and has this particular form here. So that's the passive part that tends to relax the interface. It's a fluctuation, and the interface relaxes to flat or tries to due to tension and gravity. The active forces give you sort of like a driving term, which has a more complicated expression. What it really says, remember, this sigma is my active stress, and it says that the active stress throughout the bulk, because Y spans the entire bulk of the fluid, the active stress throughout the bulk sort of determines this driving force that determines fluctuations of the interface. So this equation is very similar to the one I will write in equilibrium, where the driving will be thermal forces, but now the driving are these active forces, which are not necessarily delta correlated in time and are determined by the active stresses throughout the bulk. We then make a very simple model for the correlation of these active stresses, and we assume that it decays exponentially, and this model is suggested by numerical measurements of the spectrum of velocity fluctuations in the active fluid. And so we assume it decays exponentially on a correlate with the time tau8 that describes the relaxation of these active flows. And it has a spectrum here, which is essentially the energy spectrum of active kinetic energy, the spectrum of active kinetic energy in the system. which we can independently calculate from simulations and measure in experiments. And it has this characteristic shape, which has been actually found before, whereas small k is actually, and the crossover is really determined by the fact that the small k dissipation is controlled by friction and the large k is controlled by viscosity. And so the crossover is actually at a characteristic length scale that controls the interplay between viscous dissipation and friction, which is essentially the length over which flows generated by the active stresses extend or are screened by dissipation in the fluid. So a simple way to think about this is that you have these vertical active flows in the bulk that extend the length L eta, LV, I guess, and push up the interface. And the interface is then tends to be restored to flat by passive forces like tension and gravity. And that the interplay between these two determines the fluctuations of the interface. And then we can calculate the equal time fluctuation spectrum. And our analytical expression, actually, I realize I didn't show it here, our analytical expressions reproduces very well the simulations and qualitatively reproduces also the experiments with this characteristic crossover, although the exponent we have theoretically is a bit different, is this -6 and this is -5. So basically what this shows is that indeed that we have a way of understanding and modeling the driving force due to activity that drives these fluctuations. And it's very different from thermal driving, because thermal driving is always delta correlated in time, while this is correlated both in time and in space. Any questions about this? Okay, but more interesting, yes, go ahead.
How it is dependent on the fluid?
On what?
How it is dependent on the medium. If we change the viscosity of the medium, you have a crossover which is dependent on the viscosity. But
so the crossover that depends on the viscosity is the crossover.
What about the power?
Energy.
What about the power? This small k, will it change with the viscosity?
No, the power does not. So the location of the crossover in k will depend on the viscosity. And if you increase the viscosity, I think you move it to smaller k. But the power, no, don't change, don't depend on the viscosity, on the value of the viscosity.
Okay, so if we change the medium to air, let's say, so will it not change the system overall?
Well, okay, our calculation, it's a bit of a cheat here. Our calculation, this gets pretty lengthy calculation. So our calculation was done assuming the viscosity of the two medium are the same. You see, we have the same eta here.
Okay, okay.
Okay. Now, that's certainly not the case. Well, in the particular experiments, these are two polymers. The viscosity are probably not all that different, although active stresses actually also change the viscosity. So you should really worry about that. If the medium is air, there is actually a difference. Now, what we have also done is solve for a viscous fluid with vacuum. And that only changes some factors, like factors, numerical factors in the expression. It doesn't really change anything. But two fluids of very different viscosity, I'm not completely sure.
Okay.
Okay. Anything else?
Yeah, based on what Professor Pallab asked, I have like one question. When these experiments were performed, other than the known parameters like viscosity and say surface tension, like what all things were actually measured to correlate with the model in the experiment? Like,
say it again. What is actually measured? Did you ask what is actually measured in the experiment?
Yeah.
They actually measure the spectrum of, so in this here, what is actually measured, maybe we can go to this image. So they actually visualize the interface. They use, image analysis to calculate the deformation in terms of the angle. They actually calculate the angle, not the height, but the two are related. They calculate the angle as a function of the position along the interface. They Fourier transform, And then they measure the magnitude square of these of the Fourier transform of the angle, which I call the spectrum of fluctuations as a function of wave number.
Okay, okay.
And they also do this as a both function of, as we'll see in a minute, as a function of position and time. And then the Fourier transform both in space and frequency to measure the dynamical spectrum.
Okay, okay.
Okay?
Yeah, thank you.
Okay. Okay. Okay, so in fact, even more interesting is what happens when one actually looks at the dynamics of this interface, as a matter of fact, because then by measuring the structure, what I call the structure factor, which is essentially the Fourier, the spectrum but in space and frequency, and measure the way I just described. And what is plotted here is the intensity of the structure factor. The vertical axis is frequency, the horizontal axis is K, is wave number. And what you see, the intensity is largest at the red. There are kind of these shoulders. So there is, of course, a strong peak at zero frequency and zero wave number, but then there are actually peaks at finite wave number and finite frequency. What this indicates is correlations that you can actually see here in this sort of chymograph of height fluctuations. Again, now this is position, so this is real position, this is position and time, real space, real time, and this is wave number and frequency. If you look at the height fluctuations, you see sort of some correlations in both space and time, suggesting that, and then when you go to Fourier space, you see these peaks at finite frequency and wave number, which suggest essentially traveling waves. Now, you know that on the surface of, say, water, you can generate traveling waves, and on the surface of thermally fluctuating interface, there are similar waves at a smaller scale, which are known as capillary waves. And you see this kind of traveling waves on these interfaces as well. Now, this is surprising because now let me remind you about capillary waves, which are sort of the short scale analog of traveling waves on the surface of a lake, say. Because capillary waves arise from the interplay of tension and inertia. Essentially, and I just told you that in this system, inertia is negligible. We use the Stokes equation to model them. So essentially, capillary waves arise in the following way. You have, again, that the height fluctuation is, the rate of change of the height fluctuation is the velocity with which the bottom fluid pushes up at the interface. And then you write the Navier-Stokes equation for the velocity, say, of the bottom fluid. And here I've only kept inertia and pressure gradients, because this is just a passive thermal fluid. I could include viscosity, but I'm just trying to make it as simple as possible. You have incompressibility. And of course, there is a pressure jump at the interface, which is determined by the tension forces associated with interface fluctuations. When you sort of combine, you can imagine combining these two equations, or you have to solve them because of the boundary condition, but essentially when you combine these two equations and you can write a single equation for the interface, that now becomes a second order time, contains a second order time derivative, and so becomes actually a wave equation. And so what you get at the surface is traveling waves with actually a dispersion relation, which goes like wave number to the 3f's, and where the speed is controlled by the interplay between, I think there should be a G somewhere here as well, but between surface tension and gravitational restoring forces. No, sorry, there's no gravity here. Surface tension and density, which controls the inertial restoring forces. So capillary waves are well known at the interfaces, but they are determined by the interplay of tension and inertia. Well, our fluid is one where inertial effects are negligible. So what controls these capillary waves? And you can actually understand the occurrence of these waves by essentially decoupling between interfacial fluctuations and liquid crystalline degrees of freedom, the following way. So this image is supposed to be an image of, is an image of our simulations. Here is the interface. And what is supposed to highlight is that the filaments actually tend to orient parallel to the interface. This is actually known, has been studied before, has been called active anchoring. Due to the behavior of these active stressors at the interface, you have sort of a layer where the filaments tend to be aligned with the interface. So we can then use this knowledge to construct sort of a simple phenomenological model for the coupled dynamics of height fluctuations and deformations and orientation of the filaments just at the interface instead of without taking into account the entire bulk. For the height fluctuation, again, we know that there is essentially the balance between pressure or stress jumps at the interface and the tension. Sorry, the tension was sigma, and now here I realize I called it gamma. Sorry about that. This is the tension at the interface. And using that, you can write, well, the equation we actually have written before. We had this equation of motion for height fluctuation with the part that is the passive relaxation and this active drive. But now I've greatly simplified. I calculated the active drive just at the interface, and it turns out to be proportional to activity and essentially gradients of this angle theta, which describes the filament orientation. So I'm working here in Fourier space, but this is clearly is a gradient. So that's the equation for the height. Now I want a coupled equation, another equation for the angle fluctuations of orientation of the filaments at the interface. To do that, I go back to my liquid crystal hydrodynamic equations, sort of a simplified form. And I note that, so the first term, instead of writing the equation for the full Q tensor, I just write for the angle relaxes with a passive relaxation time. And this term decouples flow to orientation, precisely vorticity to orientation, can be sort of calculated right at the interface, and essentially it gives you a term that is proportional. It says that the angle deformations are driven by gradients of the height, okay? So height relaxes through tension and is driven by gradients of the orientation of the filament, proportional to activity. The fluctuations in the orientation of the filament relax passively and are driven by height fluctuations. If you combine these, so I've written the two equations here again, and now we can kind of understand how these waves come about. It's a phenomenon that we call delayed anchoring. So essentially, imagine you have a fluctuation in the height, and that also is associated with a fluctuation in the orientation of the filaments that like to be aligned with the interface. This orientation is what actually drives the height fluctuation that then tends to relax due to surface tension. As the height relaxes, it generates vertical flows denoted by these black circles here that in turn tend to suppress director deformation, tend to restore a flat director field, tend to restore a flat interface and a flat director field. And there is a feedback between the two phenomena, but the two phenomena occur on different time scales. Height fluctuations relax due to tension, and liquid crystal orientation fluctuation relax with this time scale tau. And so there's a delay between the two, and that's what leads to these traveling waves. Another way to think about it is that you can combine these two equations into a single second order in time differential equation, which would be a wave-like equation, and you can see how wave arise from this feedback. We can, of course, calculate the dispersion relation for these waves, and this is shown here. The blue is the imaginary part that describes the speed of the wave, although this is not linear in the wave vector, so it's not really quite like sound waves. And if we calculate numerically from our simulations the dynamic structure factor. We see this is an intensity plot. The intensity is large in this red region. And so you see that the maxima really follow the spectrum of these that we find in our calculation. So there is essentially the traveling waves only occur above some characteristic wave vector and have this type of dispersion relation that eventually becomes really actually there's a region here where dispersion relation is rather flat, independent of wave vector, but in the region near the onset, and actually these dispersion relations have been measured both in experiments and simulations, and they're very hard to measure in the experiments, but in the simulation you see quite clearly that the wave only exists above a characteristic wave vector, and in the region of onset you can actually define a speed, which would be the slope of the dispersion relation, that increases with increasing activity. These three curves are for increasing activity. So what we have here is a sort of activity-driven capillary waves at the surface of these active-passive interfaces, which are determined not by the interplay of tension and inertia, as in passive systems, but by interplay of tension and active stresses, essentially active forces. Any questions about this? Okay, now one more thing I would like to show you are the wetting properties of this system. So we know that, well, what is wetting? Wetting controls how fluids spread on interfaces. And for instance, well, we know that these are water beads on a leaf, and the cell aggregates actually can behave more like a water bead on a leaf, but they can also spread out and on the interface and sometimes even cover the entire interface. So the way we understand how a liquid drop spreads of an interface, which is the phenomenon of wetting, is in terms of the balance between the tension forces between the liquid and the interface, the solid, medium, and say the air over here, and the liquid and the air. And in principle, we have three tensions, liquid, solid, but I have incorporated that into addition. And the angle that the droplet forms, or the wetting angle, is determined by the balance between forces. So between here, this adhesion force between the drop and the interface, and the tension between the liquid and the air. And when you have an angle like this, for our droplet over here may have an angle that is actually very larger than pi over 2. An angle like this, which is smaller than pi over 2, will be called partial wetting, and a lot of liquids behave this way. When this angle becomes zero, when the adhesion forces exceed essentially the tension between the liquid and air, you can have what's called complete wetting. That's like oil in your frying pan, especially when you warm it up, that spreads and covers the entire interface. Now, these active fluids are actually quite peculiar, because not only, so now again, remember our geometry in the experiment. We have this vertical cell with the active fluid at the bottom and the passive fluid at the top. And now what I'm showing you here are 4 movies. They come in pairs. The first one is experimentally is a very low concentration of kinesin, and the simulation has a 0 activity, so it's a passive interface. And then we have one at a high concentration of kinesin in the experiments and a high activity in the simulations. So for low concentration for low activity, low or 0 activity, you see that there is wetting at the interface with an angle like that. I don't know what happened there, but anyway, I think you were able to see this. But as you increase activity, what you see that the wetting is greatly enhanced. And in fact, this fluid, literally the active fluid, climbs up the interface against gravity, generating this thin layer that eventually turns really incomplete wetting of the interface. And you see something very similar here in the simulation. So activity is pushing up the fluid against gravity, eventually leading to complete wetting of the interface. How can we understand this? Well, let's go back to what we know about how we determine the interface profile for passive wetting. So here's the geometry. Here is the flat interface will be here as 0. Here is the height profile that defines this wetting angle. And the profile is determined by the balance between the adhesion forces to the interface, this gamma W, and the tension between, say, the liquid and the air, or at least the component of that tension really tangent to the interface. So it's given by gamma cos theta, and you can balance these two forces that determines the wetting angle. But now what we have here in addition is gravity, okay? And so to find the interface profile, we actually balance this gamma cos theta, and cos theta is written here in terms of the shape of the interface. The x means derivative with respect to x, so two derivatives here, one derivative here, is balanced against gravity. For small deformation, you can neglect this derivative in the denominator, and then you can just solve. And of course, you get an interface profile that is exponential with a length scale, which is our capillary length, determined by the interplay of tension and gravity. So this will be an exponential profile, and the deformation caused by the wetting decay very quickly. In our active system, both analysis of simulation and experiments show that the profile of the interface actually is still exponential and decays with a length which is pretty much controlled by the capillary length. So this observation allows us to construct a very simple phenomenological model, which is the following. So here is my interface again, but now we have activity, and we have down here our active microtubule system. That if you see here is simulations, the microtubules also tend to form a thin layer aligned with the wall. And this thin layer aligned with the walls will cause extensile active stresses that will push up the interface. So it's as if there was an additional tension that I call gamma A, proportional to activity and to the thickness, which is a parameter here of this aligned region near the wall. So we have to now add this active force to our force balance. And here I'm actually neglecting gravity because it turns out it's negligible compared to the active forces. And that will lead to a new active wetting angle that I call theta A, that determined by solving this equation. I assume that the passive tension is still given by the passive wetting angle. And I can then combine these two equations and solve for the active angle theta A, and I'll show you the solution in a minute compared with the simulations and the experiments. And so the maximum height that the interface reaches will be also determined by this angle. And we can define complete wetting as the case where this angle is simply 0. And by setting it equal to 0, we actually get a condition for the critical activity at which complete wetting can occur. We can solve for the activity which is contained in this gamma A. When we do this, we find that actually we obtain so what is plotted here is not quite the wetting angle nor the height of the interface. It's something we call the center of mass height. And it is essentially the center of mass of the layer of active fluid that climbs back up above the mean value of the flat interface. So is the average is the center of mass of that thin layer of fluid, which we can both measure in the experiments and calculate in the simulation. So when we calculate that center of mass with this little model I showed you in the previous slide, we get this red line, which actually corresponds quite well to the points which are calculated in the simulation. And this is the point where complete wetting sets in. And similar, so this tells you that the layer increases in size with activity. So the fluid climbs higher and higher with activity. And something very similar is seen in the experiments with the Kane, a similar profile for the increase of the center of mass with activity. One eventually even sees that this profile spreads out kind of droplet of active fluid that climb up your interface. So it's really a kind of remarkable phenomenon. Let me skip that slide. So I will stop here. I'm not really sure because I don't know what time I started. Oh, there is something in the chat. I should have paid attention. Okay. I'll get to this in a minute. Maybe I'll finish. This is my last slide, so let me just finish, then I'll read the question. It's pretty long. Okay. So I will stop here since I think I probably have spoken for about an hour, although I'm not exactly sure at what time I started. And basically, I try to tell you about some of the interfacial phenomena that we've been seeing and modeling in these active-passive mixtures. Demonstrating a link of this microtubule surface alignment to these active ways we controlled and how wetting can be also controlled with activity. What I didn't tell you is something very interesting which we're working on now is about the coarsening dynamics, meaning if you actually do the experiment in an horizontal geometry where gravity is not important, then you can actually look at the kinetics of phase separation, how the system coarsens, starting from an initial quench by making droplets. And this is actually shown in these images here, where going down, the activity is increasing. As you can see, the phase separation is very different at high activity than it is in the passive fluid, where of course you form the passive fluid, your droplets are round as you expect them to be. In the active fluid, they becomes a very irregular shape, and even more so when you go to high activity, where you actually find a steady state, essentially, of droplets that are continuously breaking up and joining in this highly dynamical state. And we have some understanding of this, but I'm not going to try to tell you. But the main point that I'm hoping I made here is that activity provides you really with a new tool for sculpting the morphology of interfaces, tuning wetting properties, and controlling phase behavior. And it might allow us to sort of really create new active materials by doing this. So with that, I just wanted to put up again the faces of the people who did all the work. And thank you all for listening. And I will look at this question in the chat while I wait to see if there are other questions. So it says, for a bacterial suspension system, does the concentration gradients of availability of microbial growth limiting nutrients affect the dynamics and morphology of bioactive interfaces? Yes, people have studied these as well. And I think the answer is yes. There is, for instance, a paper by Alison Patterson and I think Paolo Araccia some years ago, where they actually study precisely the problem, and they see that in that case, the interface also advances. There's also, they're interested in that case in the dynamics of the interface. Then he says, is the interfacial fluctuation somehow related to the chemotactic tendencies of the bacteria? Again, I think so. I can, if you send, and is the active stress impacted in any way by the external hydrodynamic fluid velocity for a flowing system? I am not sure about the answer to the last question. I'll tell you something about the last question in a minute. Concerning the first two questions about interfaces between in growing bacteria and the effect of chemotaxis. I can refer you to this work I mentioned, and if you send me an email, I can send you the reference to the paper. About whether active stresses are impacted by external hydrodynamic flow, that's kind of interesting because that relates to the question about, suppose you shear an active fluid, what is the shear viscosity? You can actually show that in bacteria that like suspension of E. coli, if you shear an active fluid, you can actually, the swimming bacteria can sort of act promoting flow, and therefore you will actually measure an effective viscosity, which is much lower than the viscosity you would measure if your bacteria were dead. In fact, you can even measure zero shear viscosity and negative shear viscosity. In fact, people call this a superfluid. Of course, it is not. So in other words, yes, active stresses can strongly impact the response to externally applied flow. And that's another very interesting area of study. Anyway, are there any other questions?
I have a question.
Okay.
So
Is that Abhinava?
No, Harinatha, Harinatha, your voice is breaking. We're not able to hear you. Probably you can type your question.
Okay, maybe while Harinatha types, you can have a question from Abhinava, who has his hand raised.
yeah, can you hear me?
Yes.
Yeah. So yeah. So I'm asking, like, initially when we're seeing, like, we're modeling the experiment, we considered the frictional energy due to the, like, substrate and also due to the walls. Like, then when we move to the wetting phenomena, we see, like, with the increase in activity, there is an increase in also the wetting. So I believe, like, there will be also an increase in the frictional force and the energy. So are we taking that into account, or like it has been taken into account by any other factor?
Yeah, well, so when I talk about friction in describing the interfacial fluctuations, what I have in mind is the following. So this is a thin cell, about 60 micron thick and centimeter in the other, millimeters in the other direction. And so what I'm thinking that friction comes from is friction with the side walls. So I'm really doing sort of a lubrication approximation of the hydrodynamic equation for a thin layer. And that's where the friction that I have in the Stokes equation comes from. Okay. When I talk about the increased force that allows the things to climb the wall, I'm talking about adhesion to the thin side wall. So again, here is my, I don't know if you can see my hands, but here is my cell, right? I'm talking about adhesion with the thin side walls over here. And actually, experimentally, they are trying to think about treating the side walls so that, although they haven't really done this yet, they can actually alter the anchoring properties and see of the pneumatic, of the liquid crystal. Right now, the liquid crystal tends to align with the side walls, but you can actually treat them by change. We have done this in the simulations, of course. We can actually change the boundary conditions and not allow to align and see how that changes. And if you put in boundary conditions that require the filaments to be perpendicular to the walls, you strongly suppress this active wetting. Okay. So I don't know if I answered your question, but.
Yeah.
Okay.
Thank you.
Okay, I can read the question in the chat now. Wait a minute. There's a question in the chat. It says, I've been following some of your older papers where you have used Langevin equations and shown phase segregation. Could the same have not been achieved for the segregation case shown here? What made you consider this model, which is closer to a phase field model and Langevin model? Well, so I think the older work you're referring to is on what is called motility-induced phase separation. And that is a phenomenon where if you have just one component system of purely repulsive spheres that are self-propelled, they phase separate due to due to motility, even though there's interaction is just repulsive. Now you can write a continuum model for that system as a sort of a phase field model. The difference here is that we have coupled the phase field to the nematic degrees of freedom, and that coupling is important to understand what's happening in this system. So yes, I think here we really needed to couple. So that was sort of a, if you write a continuum model for the system you're referring to, you have just a scalar field, but here you have coupling not just to flow, but also to nematic degrees of freedom, which is important. I don't know, Siddhant, if I answered your question. Okay, we also have a question from Rika or Richa.
Richa, Professor Richa.
Richa, thank you.
Thank you, Cristina. It's a very, very nice talk, and like most of the part is not directly related, but like many related to my work. So I have one simple question. So you said about the traveling wave. Can you hear me?
Yes, not very well. That's why I'm trying. I'm kind of trying.
Oh,
yeah.
Okay.
Go ahead.
Okay. Sorry. So you spoke about the traveling wave. So you know that dictyostelium, the cellular slime mold, also generate the traveling wave, which is like active traveling wave of CAMP, right? Some chemical traveling wave.
Okay.
So I'm just like asking, is it like what is the difference? Not what is the difference. Is there any difference in the order of magnitude of that traveling wave by the traveling wave which is generated by your active matter?
And by order of magnitude, you mean what, like speed?
Yeah, like speed, time, and the like.
I don't know. The short answer is I really don't know.
Okay.
Yeah, I'm not really sure. You're talking about waves that are generated by sort of couplings. In what system are you thinking about? Which system are you thinking about?
Yeah, I'm talking about the cellular slime mold dictyostelium discoidea.
Okay.
Yeah, so they also generate some traveling waves.
Yes, sure. In many biological situations, you generate traveling waves.
Exactly. Yeah.
Coupling
so that is
mechanics to chemistry, essentially.
Yes. So I'm talking about that, like in the real biological system, whoever generates the traveling wave, what is the difference between this active matter generated traveling wave?
Quantitatively, I don't know. Qualitatively, I think the main point is that it, in some sense, is similar in the sense that, certainly from what gives the traveling wave is always whenever you have two quantities or two fields that have a purely relaxational dynamics, but they're coupled and there is a feedback between the two. So in that case, probably some mechanical, some mechanics couples to chemistry that feeds back into changing the mechanics. And here is height relaxations coupled to orientational fluctuations that feed back into driving the height fluctuations. Whenever you have this feedback, essentially, then you can always generate traveling waves. Okay.
I got it. So it is very easy to incorporate your model in that kind of system.
Yeah, it's a very general way of generic way of getting traveling waves, I would say, yeah.
Okay. Thank you.
Sure. There is a question from Harinad in the chat. Let's see. My question is on the wetting due to the activity. This kind of phenomena is also observed for chemical releasing Marangoni bacteria, Bacillus subtilis. In that system, the climbing of the fluid is occurred due to Marangoni flow. I assume there is no such Marangoni kind of flow in your system. It is happening purely due to activity. How different is your active system for bacterial biofilm? Thank you. Don't know. That's very nice observation. I don't think we have Marangoni effects here. But I would be very interested in, I don't know this work. I'd be very interested in seeing it, and would very much appreciate if you were to send me a reference. There is a question from Udit.
Hi, Professor.
Hi.
So I just wanted to go a little bit more into what you are explaining. So instead of a concave interface, which for the wall climbing experiment, suppose if the solid surface is such that you have a convex interface, does the dynamics change? You still have the wall climbing and the droplet formation, or does it remain the same?
We looked at this. No, I can't. So what you're talking about is changing the passive wetting angle, right?
Yes, I mean that is a solid fluid. So you have basically a concave.
Right. I have a yeah.
And now you have imagine that starting is a convex because the solid I've changed the solid wall.
We actually looked at this numerically, and I cannot exactly remember. My recollection is that it remains the same, but the effect is weaker.
Okay, because your initial condition is weaker. And the second question is, if the fluid on top is active instead of the fluid on bottom, do you have the same effect of like the wall climbing, or is it like going down? Like let's say the top fluid is the active fluid and the bottom fluid is the passive fluid. So does now the interface goes towards the vertical?
I would imagine the effect is the same, because the gravity is really negligible here in the context of this effect.
Okay. Thank you. Thank you. Yeah. Thanks.
Sure. Okay.
There is one more question before by Chaitanya. Can you please comment about the role of topological defects in this system? In the chat box before Harina, there is one question.
I see. Well, we haven't really looked at this. There has been work by Julia Yeoman's group looking at the role of topological defects in advancing at the interface of an active nematic on a horizontal geometry. And it could be that you can interpret these, let me call them pushing forces on the interface in terms of defects, meaning you certainly have defects, that defective type structure that are sort of the +1F defect in particular, which is like the one that looks like the comet that hit the interface with their nose and push the interface upward and then turn and slide along the interface. So it might definitely be possible to describe these flows, these phenomena in terms of defects. We actually haven't done that. I should say that one of the reasons we haven't done that is that the active fluid is in the microtubule density in this particular active fluid in the experiments is low enough that you cannot really think of the active fluid as being in a nematic phase, but being in an isotropic phase. There is local nematic order generated by activity, but without activity, the fluid will be isotropic. Then the microtubule system will be isotropic. So that's something we are, thinking. We are thinking of looking at the role of the defects in this system, but we haven't really done that yet. Anything else?
Any other questions?
Okay, well, thank you.
I think there are no other questions.
For listening, and
thank you. Thank you for this nice talk. Yeah.
Thanks a lot. Then I will stop sharing, I guess. And okay, thanks for coming late, so late. I guess it's pretty. What time is it? Is it now?
It's 9 p.m.
9 p.m. Okay, it's not too bad. So okay. Now, instead, I guess I'll go have breakfast. So,
yes.
Goodbye, everybody. Then, bye-bye.
Okay, bye-bye.
Bye.
Thank you. Thank you, everyone. Bye-bye.